Chapter 3, Topic 3.6, Prokaryotic Classification Systems. In this topic, we go over how we currently classify prokaryotes. We're going to talk briefly about Berge's manual, taxonomic schemes, and species and subspecies in prokaryotes. At the end of this recorded lecture, you should be able to name 4 divisions of prokaryotes ending in cutes and describe their characteristics and explain what a bacterial species is. Classification systems are quite common in science, and microbiology is no exception. It helps researchers and clinicians identify and study bacteria. Berge's Manual of Systematic Bacteriology presents a comprehensive view of prokaryotic relatedness, combining phenotype, which is shape and that kind of thing, information with ribosomal RNA sequencing. We're going to talk about that more when we get to DNA replication in prokaryotes. It is most up-to-date compilation of data, and it is constantly being changed and updated. Berge's Manual of Determinative Bacteriology is based entirely on phenotypic characteristics. It is the workhorse of identification. The rRNA sequences may be more accurate, but the phenotypic characteristics have been used for longer, and most bacteria that we're able to grow in the lab are characterized this way. On to taxonomic scheme. Berge's Manual of Determinative Bacteriology organizes the prokaryotes into 4 main divisions based on the cell wall. Gracilicutes have gram-negative cell walls. Gracili refers to something being delicate or thin, like a thin cell wall. Think of graceful. The firmicutes have gram-positive cell walls. Firm, thick. Get it? The tenoricutes lack a cell wall. Tenor is kind of like tender. The mendosicutes are the archaea. They have unusual cell walls, meaning they don't have peptidoglycan in them. The gracilicutes and the firmicutes have the greatest number of prokaryotes that have been characterized to date. Now let's talk about species and subspecies in prokaryotes. For multicellular organisms like animals and plants, a species is most often defined as a population of which the individuals can successfully reproduce with each other. This works. Horses can breed with horses and have fertile offspring. Horses and donkeys can breed, but their offspring, mules, aren't fertile. It makes everything very easy to characterize. Unfortunately, we can't use this definition. It won't work with bacteria and other prokaryotes. They don't reproduce sexually. They exchange bits of DNA with very unrelated individuals. It's all very messy. So, we define prokaryotic species as a collection of cells that share an overall pattern of traits. They should share 70 to 80% of their genes. So, all E. coli are the same shape, coccobacilli, size, and we should find them in the same habitats, mammalian guts rather than in hot springs, and with approximately the same metabolic characteristics. Because of the 70 to 80% variation, individual cells will have some differences in characteristics. These individual cells can reproduce asexually or make clones of themselves and form what is called, in these lovely colonies here, variously a strain, a subspecies, or a type. You may already be familiar with different strains of bacteria. Most E. coli are harmless and actually help you out by living in your intestines. The hemorrhagic kind of E. coli, known as H10157, it produces a toxin that causes bloody diarrhea, or in the most severe cases, kidney failure and death. That's pretty much it for this topic. I do have a Khan Academy video that is a beautiful summary of Chapter 3. So go ahead and watch that if you feel like it. You should now be able to name 4 divisions ending in cutes and describe their characteristics and explain what a bacterial species is.